Now, welcome back. Actually, in the first video, the ART was not completed. So, we will resume the next part of the ART. We have discussed about the advantage of the in vitro fertilization, in which the fertilization is confirmed before the implantation can occur and gives women with the damaged oviduct the opportunity to carry their own fetus. It means the woman which has blocked fallopian tube or narrower fallopian tube, by the due to which the female gametes are not transferred to the towards the uterus through the fallopian tube. Now, those type of women become pregnant by the help of this technique. Here is the disadvantage. Disadvantage of the implantation in the uterus does not always occur. Actually, when we transfer the developing embryo in the uterus for the implantation, it is not necessary the developing embryo is getting transferred or getting implanted in the uterine wall. It is a matter of chance only. High risk of the twins or triplets, which also increases the risk of the complications and miscarriage. During the process of embryo transfer, we transfer more than one embryo to the uterus of the female. So, there is always risk of the implantation of more than one embryo to the uterine wall. And when the chances of twins or triplet occurs, it causes always complications in the pregnancy and it increases the risk of miscarriage also. Side effects associated with the fertility medications. There are so many side effects, hormonal side effects, getting reflected in the mother's body. So, these are the different disadvantages of the IVF technique. Now, the normal results lies up to 27 or 25 to 35% in the IVF. Look here, this animation of the IVF, how IVF is being done. Look this animation. It is a normal process. Ovulation is getting occurred from the graphene follicle. The secondary oocyte is released and travels through the fallopian tube. Here inside the fallopian tube, the different sperms are coming. These are getting attached with the surface of secondary oocyte. It is a normal process. In the assisted reproduction, what will be happen in vitro fertilization, IVF? Look this animation. Here the multiple ovulation takes place due to hormonal treatment. Look, this needle is gone through the vaginal canal, reaches to the ovary, sucking of graphene follicle, secondary oocytes, this needle is back. Now, it is the female gamete kept in the petri dish. The mature sperms are kept along with it. Where is the secondary oocyte or female gamete? After the fertilization, the embryo is getting developed, and this developing embryo is further transferred to the uterus of the mother. It is released inside the uterus through cervix. This developing embryo will get implanted in the uterine wall, and then pregnancy was caused. Here it is the result of the in vitro fertilization. So it was the animation of in vitro fertilization. Now the second ART technique, that is the zygote intrafallopian transfer, ZIFT. ZIFT is also called as tubal embryo transfer. Actually it is similar to the IVF. Fertilization occurs in the laboratory, and then the very young embryo is transferred to the fallopian tube instead of the uterus. It is the basic difference between the IVF and ZIFT is that in the IVF, the developing embryo is transferred directly in the uterus, while in the ZIFT, the developing embryo in very early stage is transferred to the fallopian tube of the mother. This is the difference between the ZIFT and the IVF. There is the GIFT or gamete intrafallopian transfer. It involves transferring eggs and sperms in the woman's fallopian tube. So fertilization occurs in the woman's body. We are transferring the egg as well as sperms both in the fallopian tube of the mother, and the fertilization will take place naturally inside the fallopian tube. So it is different from the ZIFT or IVF. In which case GIFT will be followed? When the distal end of the fallopian tube is blocked, means the secondary oocyte cannot getting entered inside the fallopian tube through fimbriae. In that condition, GIFT will be followed. Now the intracytoplasmic sperm injection, ICSI, it is often used for couples in which there are serious problems with the sperms, when the sperms have different problems. Sometimes it is also used for older couples, for those with failed IVF attempts. When the IVF is not done successfully, in that case, in the older couples, the ICSI will be followed. In the ICSI, a single sperm is injected into a mature egg. In the previous three conditions, in the in vitro, in the ZIFT and in the GIFT, a sperm was naturally entered or naturally fuses with the female. But in this case, the single sperm is injected in the female egg, and inside that female egg, the nucleus of the sperm get fused with the nucleus of the female gamete to perform the process of fertilization. And then this embryo is transferred to the uterus or fallopian tube, either like ZIFT or like the IVF. So it is the ICSI. So these all are the ART techniques by the help of which we can help the infertile couple. Thank you.